of a live stream that I made in the run-up to the 2025 exams. But you can follow along with me as I spend a few hours filling in this A3 poster. As it's a live stream, I respond to questions given to me by hundreds of real students as they prepare for their exams. I have 7 of these live streams that I've re-uploaded, and over the course of this year, I'm going to host the remaining live streams and re-record the old ones so you've got a chance to take part. If you want to join me, then please subscribe so you get notified when I announce one of these live streams. It would also be great if you could subscribe because I just hit 94,000 subscribers. So pretty close to that 100k milestone. So if you're finding my stuff useful and want a free way to help out the channel, that would be amazing. Now, I don't do sponsorships for things like VPNs, but the other things I make are entirely focused on helping you get the best out of your A-level. And selling all these things helps me make the free content on YouTube. You can get this poster and posters for every unit, both completed and filled in as digital files and now as a revision book. You could also follow along with each of my main videos with worksheets. These are designed to give you an active role and make the information sticky. I also have an app for you to test yourself, and I let you use all the paper 1 for free without ads before you decide if it's worth unlocking papers 2 and 3. Altogether, it's thousands of pre-made quiz questions and flashcards. And I have over 17 hours of exam question walkthroughs, as well as lots of other resources over on Patreon. And starting now, I'm going to be doing an exam question focused live stream most weeks that are exclusive to my Patreon supporters. Thank you for listening to all of that. Let's get started. We are starting, everyone, with divisions of the nervous system. No, you cannot get a fight or flight is not something that we would ever expect a 16 marker on, or you can only really go off what we've seen before, and that's just not it. Right, division of the nervous system. What is it, please, everyone? Please just give me a definition of what the division of the nervous system is. Don't need to go into a lot of depth about the bits, but what's the definition? Okay, so it includes the CNS and PNS. I was after more of a kind of a general. We're going straight into the different sections, but I am going to one more general definition. Okay, got some good stuff there. I think you kind of got it. Right, I have put, here we go, the human nervous system, a body-wide system of nerve cells that collect information from the world, processes, then takes action by directing the organs and muscles for the transmission of electrochemical messages. So just a general definition of what is the human nervous system. It is this large collection of nerves that have certain functions, bringing information in. Obviously, we've got the CNS, which processes that information, and then it is able to redirect that information and kind of shape the world by moving its body and all the rest of it. So that is how we interact with the world, experience and interact. And fundamentally, we'll go into this obviously in a lot more detail. This is through electrochemical messages. So let's break this down. What are the 2 main divisions of the nervous system if we're going to separate it out? I know some people have mentioned it, but just go for it again. Okay, Rose is in there first from after I answer it, and Jenna was quickly behind. Loads of you going over there. So we've got the CNS and PNS. I will give just a little bit of, I'll just give the details for these first, and then we'll maybe go into more details about them later. The central nervous system, complex processing. The peripheral nervous system. So we've got messenger neurons across the body. We have 2 different ones though. We have sensory, which are afferent, and motor, which we called afferent. I probably should have asked that as a question, but oh well, it revealed it pretty early. So please tell me then the 2 divisions of the central nervous system. Okay, we're all good there. So lots of people there with brain and spinal cord. Thanks very much for that money motivation. That's really kind of you. So brain and spinal cord. I won't keep this hidden from you because I think it's a little bit simple. The brain obviously, we're going to summarize what is the brain. It seems kind of obvious. All conscious and most unconscious processing. Seems a bit weird to say most unconscious processing, but we do also have the spinal cord as well. And the spinal cord will receive and transmit information, but it's not just a receiver and transmitter of information. As we'll go into in a second, it also is able to do some very, very basic reflex processing. So not necessarily all of the complex processing is in the CNS. Yep. Okay, go careful how you say that as well. I don't know. Okay, so let's go into the peripheral nervous system. Please break that down to me. What are the two divisions of the peripheral nervous system? Okay, lots and lots of you there. I don't know who got there first. Somatic and autonomic. Brilliant. Okay, let's break down first this. What am I going to reveal? I'm going to reveal both of them, actually. Let's break down the somatic nervous system. What is that? I realized money motivation didn't thank you properly for that little financial donation you just gave in the chat. I do appreciate it. But, anyone else watching, don't do that. I'd rather you actually get something out of your money. So, get a worksheet or something like that. But it was very kind of you. Yeah, so voluntary skeletal muscle control is what a lot of you are saying. So voluntary, I'm loving seeing the voluntary element in there because it does make the distinction between what the other section is, the autonomic section. Excellent. So we can leave that there because it doesn't break down into any other sections. But we do need to go into a little bit more detail about the autonomic system. So tell me what it is, but then tell me the two sections as well. What are the two sections of the autonomic system? Here's one. Yeah, really good. Okay. So involuntary, including heartbeat. Kitty controls involuntary movement. Yeah. Unconscious control is definitely an element of this. We don't choose any of these things. So we have got autonomic nervous system, control actions of internal glands. It is an involuntary system. Somatic control skeletal muscles is a voluntary system. All right, let's break down now the autonomic system. So first of all, what are the two terms? Hopefully we can get that right, and then we'll look at getting these the right way around. JSL Scoot can pretty much read my mind because we've got, and Jasmine was the first person to say one, and when I said it, parasympathetic and sympathetic is correct. So let's get this the right way around. What does a sympathetic system do? So what's happening with the sympathetic system? Okay, we got loads and loads and loads of people talking about fight or flight. A couple of people saying rest and digest, but that's not the right way around. But it's kind of tricky to remember. I don't know if anyone in the chat has a particular interest in way they remember which way around it goes. So yeah, sympathetic, for those of you who have typed it out, you could do the opposite, which is parasympathetic. So sympathetic, heart rate increase, increased sweat, increased breathing rate. Don't know if anybody mentioned release of adrenaline, noradrenaline. Excellent. One or two. Actually, who said that? Lola, well done. There were a few of you as well. So we have parasympathetic. So lowest heart rate, lowest sweat rate, lowest breathing rate, and we have acetylcholine release. I don't know if you necessarily want to get much detail. Depends on the question. Maybe not. But then we have the opposite there, which is rest and digest. What is the final box, everyone? If you can say what the final box is, and maybe give me a little definition. Yeah, rectum constricts. It certainly does. Yeah. Homeostasis. Great, Leo. Really nice and fast. And Shadow, Alicia. Not possible to come. This is a very AO1-y section when we have evaluations here. Okay, maintaining of internal environment from Lucy. Zoe, balance between sympathetic and parasympathetic. Balance is a really good term there. So let's fill in this final box together then. Homeostasis. The internal environment is balance between sympathetic and parasympathetic systems. Okay. We now, everyone, are going to move on to another section. So I've got a little diagram here of some neurons. So in the spec, it says the structure. So we can kind of like look at it and kind of see what's physically going on. And also the function. So I'll try and do that as simply as I can with this, which is definitely going to be what? What is the structure here called? Yeah, this is called the reflex arc. It's not mentioned explicitly in the spec. It just says sensory relay and motor neurons, but it's pretty well to know that this structure itself is called the reflex arc. And I think I'm right in saying I have seen in a past paper, although I can't guarantee this one, it's been a while, that this structure is in there and has to describe it. I've definitely seen past papers where there's literal drawings of the 3 different, or at least one of the different neurons. You need to identify which one, which is really quite tricky. So we've got this, I'll reveal this actually a little bit and then just talk you through it because I don't know how it would work as a question. We have got the sensory neuron at the top, the relay neuron to the right, and the motor neuron at the bottom. So yeah, I mean, I feel it's a bit of a mean paper because everyone kind of draws it a little different. So I don't know. So then we got those 3 neurons, and then we've got some other little bits there as well. Sensory receptors, myelinated axons, the synapse, the dendrite, and the effector, and we'll all kind of go through that. So this is which, like, what do we need to be aware of? It does explicitly say in the spec we need to be aware of the structure and function of neurons. How much detail that means is kind of up to AQA. Often people go, "Oh, can this question come up?" And the reality is AQA reserves the right to ask anything that they've put in their specification however they want. And then all teachers have to make the decision to prepare their students the best they can. That means you have to make a decision about how much depth you go into. Sometimes decisions are right, sometimes they're not. They're not right, but we'll see. So I think I get the balance right, and I find out every year if I do or I don't. But I, like every other teacher and content creator, makes my best decisions I can. So there is the diagram. Let's go into the process then. Let's go into the sensory neuron. What happens at the sensory neuron? So detect sensations at sensory receptors. That's a good start, George. Or detect stimuli. Yeah. So do you text him like because I'm there's pain from mole? Yep. Sends impulses to the CNS. We could jump ahead of ourselves, maybe just a little bit. Okay, text changes in the environment. Yeah, and then Boopity Doopity and some other people are talking about the role of an action potential. Not all axons are myelinated, but in this diagram it is, and often they offer sensory. The sensory neuron detects sensations, so the example is pain at sensory receptor. Action potential travels along the myelinating axon, and the electrical signal is converted into a chemical signal at the synapse. So what I've done there is I have talked about the journey from the sensory receptor all the way to the synapse. And action potential is how the message travels along the the neuron is an electrical impulse. We spend a lot of time talking about the synapse and synaptic transmission, and we will do in this video. But as you know, some of these neurons are very, very long, like a meter long. They travel along as an electrical impulse. Do you have to know how the reflex arc works? I mean, this spec does ask you about the structure and function of the neurons. So it's not just about what they look like, but like how are they working? So yeah, that's what we're going with. Okay, so let's talk next about the next bit, the relay neuron. Tell me what the relay neuron does. Carries information, so carry information from the sensory neurons carried to the motor neurons. It's true, there's a little element, which Rosie, I guess, has popped in as well immediately afterwards, which is info to brain. Olivia, a little bit more detail, receives info from sensory and sends to the CN and immediately to the motor, and that's what we will go with for me. So the relay neuron detects a signal and forms a new action potential, sending signals to both the CNS and motor neuron. Okay, we've nearly completed the journey here. We've got to the relay neuron, to the end, back to the motor neuron. We've got a little synapse there. Please tell me what happens at the motor neuron then. I could definitely see, if it's not already been, and I'm not going to go look now, this would work so well as an applied question. Talking about somebody like burning themselves and moving their hands before they realized and then being surprised how fast they moved. And then you having to explain the function of neurons and the basis of that. That would be good. Okay. It's not that bad. I'm talking you through it. I'm talking you through it right now, Wise Club. It's not that bad a question. Okay, so we have got connects the CNS to the effectors, transmits information from relay to effector muscles and glands. So good one, Jenna there. A lot of good terminology used in the chat here. So yes, the motor neuron detects the signal from the relay neuron. Again, this is through synaptic transmission. We had that in the synapse. And this signal travels down its own myelinated axon. The signal reaches an effector. And so, yeah, there's a few different types of effector. We've got they're actually called motor endpoints, but we don't need to go into much detail, but where it's connected up to that muscle. And then the muscle contracts as a reflex and pull it away from the source of pain. But this is the weird element that people will do an action, like you'll move your hand really, really quickly, that you touch like a hot stove. And sometimes you'll be like surprised, like, why did I just move my hand? And it takes a moment to realize, oh, I moved my hand because it was really hot. Because the CNS hasn't been told yet, and the body just moved on its own, which is pretty useful, very impressive. Thanks, Sia, that's really kind of you to say. I'm glad they're really helping. All right. Nodes around via like the little bits in between the Schwann cells. For some reason, they have the electrical impulse travel faster. I don't think we need to get to that level of depth, to be honest with you. All right, guys, we are going into synaptic transmission. I know this is a bit that people really don't like, so I'm going to try and spend as much time as you need on this. So feel free to ask questions, and we'll go. The purpose of the myelinated sheath is to increase the speed of the electrical impulse. Not all neurons are myelinated, but some are. All right, let's go first of all with like what is the synapse, and also tell me what are the little dots there and what are they doing. Okay, we've got Aisha there going to our neurotransmitters. We've got Tia's in synapse, a tiny gap between neurons. That works. Nice definition by George. Process of neurons communicating by passing on chemical messages. We've got Gina there already with the term vesicles. Okay, so you label them up for me. C is not meant to be put into the synaptic cleft, but I can kind of see why you think that. Okay, So we have, just as a little definition, before we go through the process, and I really want to spend some time on the process because I think it's very confusing to people, I want to talk you through just our basic definition of what we got here. The synapse is located at the end of a nerve cell, so at the end of, of a neuron. It allows communication between neurons by transmitting chemical signals. So, a basic definition, that is what a synapse is. Obviously, there's a lot of structural things going on, but that's fundamentally what it's there for. So, neurotransmitters. These are the chemical messengers that are released from neurons. I know there's some just kind of free floating in the brain, but let's just leave it from there. Neurotransmitters, chemical messages released from neurons. These either make the development of an action potential in the postsynaptic cell more likely to form excitatory or less inhibitory. I just want to pause there a little bit because that's going to get confusing. There are different types of neurotransmitters. It's kind of like a soup of different neurotransmitters in this synaptic cleft. And whether the message is passed on or not depends on the proportion of different types of neurotransmitters. There's neurotransmitters that make it more likely for that message to be passed on. We're going to call it excitatory. And if it makes it less likely, we're going to call it inhibitory. But I'm going to go into that again in a second. I just wanted to include that in my definition of what neurotransmitters are. Okay, I'm going to give you a moment now to talk about the process in the chat. And then I'll go through it stage by stage. So just throw into the chat all the elements that you think are happening between when this action potential, which is the yellow arrow, comes down the axon of the presynaptic cell to the point where the next neuron fires. Tell me some bits, and I'll pick that up in the chat. Okay. Do we need to include summation? Yeah. Okay. Yeah, no, not really, dear. There was a question a long time ago where in the mark scheme it talked about the chemicals, and that was like it was the first year, or that was maybe the specimen papers they handed out, and in 7 years as far as I'm aware, that hasn't appeared in the mark scheme again. So yeah, I wouldn't. If I was to remake the synaptic stuff that I did, which I probably will at some point, I'll probably take that out. Because when I made it when that was relatively new. So yeah, I don't really need to go into that much level of depth. Okay, we've got someone calling a reuptake, release, vesicles. So that's giving me a full collection there. Let's see if I can see your first message. 1, the action potential reaches the presynaptic knob. Celeste said 2, stimulates the movement of vesicles containing neurotransmitters to release the neurotransmitters into the synaptic cleft at the presynaptic membrane. Wow. Let's see. That was 2. 3, the neurotransmitters diffuse across the synaptic cleft and bind to the complementary receptors in the postsynaptic neuron. And 4, excitatory neurotransmitters increase the chances of an action potential. Inhibitory transmitters decrease the chance. Brilliant. Pretty much as good of what I wrote, but different. So let me talk you through this, guys, because you did a great job in the chat altogether. But let's think about it. Okay, so I'm going to go through this with you now, and hopefully that'll help you a little bit. So what we've got in this structure is that yellow arrow is an action potential. That represents the electrical signal traveling down the axon of the presynaptic vesicle. Just zoom in here so I can read my writing. Okay, the vesicles are represented by those little bubbles with the dots inside it. So they're let's just say the little bubbles with neurotransmitters in them. So what they're going to do is they're going to merge with the cell wall, and then they're going to release their neurotransmitters into the synaptic cleft. So yeah, they release neurotransmitters to the synaptic cleft. I put that in, let's see. They're detected by those little green things that I've got on the other side. They are receptors. They're on the postsynaptic neurons membrane. So that's how the information is passed across the synaptic cleft in a very, very simple way. But whether that next cell will fire or not depends on something. It doesn't just fire the moment it detects a neurotransmitter. because there's lots of neurotransmitters floating around. If it was too sensitive to neurotransmitters, it would fire too much. So what it needs to be is it needs to reach a threshold. So it's usually like a lot lower, and the more like excited neurotransmitters there are, the closer it gets to the threshold. But if it never reaches there, it won't fire. But when the excitatory influences are strong enough, it will fire. Now, an element of this is there are some neurotransmitters that are inhibitory. That'll make it less likely for nerves to fire, and we can manipulate these with drug treatments. So yeah, all the charges that add up to see a professional has passed. So let's go drink. And then if we've got a new expression potential will form, and the message will be passed on. So I know this is a little tricky. The cell membrane is like the skin of the cell. It's the outer surface of the cell. Okay, let's see if there's any questions I can help you with. Throw some questions. If you guys have questions on this one, stick on it for a second. You good. Let's just stick for the synaptic transmission questions for now. There's some questions on some other stuff. So summation is this process. The sum, addition is a calculation of the excitatory, all the excitatory influences, and all the inhibitory. Let's say one adds, one takes away. If the end sum is excitatory still and excitatory enough, then the information will be passed on. Okay, I've got an extra little bullet point because of a question that came up a few years ago, which blew my mind that it was there, but people answered really poorly on. So I've got this in just to kind of make this clear, because there's a question like, I can't remember exactly how it was phrased, but it was like, can information go both ways in a synapse, and why? That wasn't exactly how it was phrased, but it was along those lines. And it's like, well, okay, how would you answer that question? That's what I think it was a fair few marks. So yeah, okay, in the chat, why can't, I can tell you this now, it can't happen both ways, and why? How do you explain this? No, there's no evaluation here. For a lot of these like very, very, very biological processes, it's hard to evaluate it really. Here we go. You got the term unidirectional there, because the receptors are on one side is one explanation. Yeah, the structure of it and where the receptors are. Diffusion, high to low concentration. Yeah, no receptors. We've got a lot of people saying no receptors. Also, no neurotransmitters are held in the dendrite of the opposite cell. That's so unidirectional information can only be passed in one direction at the synapse. This is due to the location of neurotransmitters, the presynaptic neuron, and receptors at the postsynaptic neuron. But yeah, also the fact it doesn't hold neurotransmitters. All right, good. That is synaptic transmission, everyone. We're now going to go on to the endocrine system. Right. So first of all, to give me a definition of what the endocrine system is. Okay, we've got glands and hormones. Collection of body-wide system of glands. Yeah, lots of hormone stuff. Okay, good. Yeah. Collection of glands that release hormones that regulate bodily functions. And keep in mind, we're talking about hormones here, but this is psychology. I know it's biopsychology, but it's psychology. We might want to point to the element that this influences our psychology as well, our behavior. So we might want to have some hormones that we think about that we can actually talk about behaviors with. So please throw in a hormone and its associated gland and its behavior or physical change. So we've got pineal, melatonin. We've got EM straight away over testosterone and the potential link to aggression. Yeah, we've got thyroid gland, we've got insulin. So what I've not done here, I've not mentioned everyone. So thinking about endocrine system questions, you guys might have seen some that I haven't seen. But I remember one a little while ago, which was like outline the any hormone and its associated gland, and it was like except adrenaline, and I was like, oof, that's mean, because obviously you're going to know a lot about adrenaline for the fight or flight response. So that was kind of interesting. But it looks like most of you have I think, yeah, George, you've seen that question as well. I was like, everyone was like, oh, I'll talk about adrenaline, and then saw the second sentence. I was like, oh no. So yeah, I'm not going to suggest that you remember every single gland and every single hormone associated, but I'll pick some, maybe some that you've used in other sections. Oh, did you, man? Did you see my exam tutorial video? I hope that helped. I think I say the same thing now. So, what might we talk about? Pituitary, master gland. We've got ACTH as the hormone, controls release for the hormones. Would I remember that? Maybe, but I think there's other better ones. We're going to need to know the pineal gland, melatonin, and the modulating sleep anyway for the biorhythm stuff. So we might as well just have that kind of ready to go. It's a gland. It's a hormone. We kind of know the function of it as well. We could even go into details about how it's triggered by the suprachiasmatic nucleus, if you remember that bit. The pancreas, insulin, regulates sugar levels, just because it's so well known. Like, you probably just know that anyway, or maybe you don't, but it's there. And then the testis, testosterone. And we've got both the physical change of the secondary sexual characteristics, but we've also got the link in the aggression unit, potential aggression. So all things that we need to know anyway. But then, of course, we do have the adrenal gland and adrenaline and the whole role in fight and flight. I reckon that's enough, to be honest. I know there's like the thyroids and stuff like that, but do we need that? I don't know. I don't think so. Okay, let's go. So yes, some from nowhere, Andrea. There are some substances that are a hormone in the bloodstream and neurotransmitter in the brain. So yeah, that's maybe why I got different terms. Although sometimes they're just phrased differently depending if you're in America or the UK, which is fun. Let's go fight or flight, everyone. First of all, what I want, let me get my notes up here, is a little definition. What's happening in the fight or flight response? I've tried to make this quite simple. I know there are past papers where this doesn't make it that simple, but then there's lots of questions where it is just relatively simple. So let's find a balance here, everyone. Let's find a balance. Oh yeah, we need just some glands. We've definitely seen questions on glands. Evolutionary response to stressful situations is a really good starter sentence key. Okay, adrenaline, and we've also got Grace's already talked about cortisol as well. We are going to go through evaluations of the things that we need evaluations for. We just haven't got to any of those sections yet. Okay. Yeah. That jump scare in my fight video was mean. I thought it was hilarious when I made it. Okay. So yeah, let's summarize this in as few words as possible. I know it's quite difficult, and I know I'm kind of like simplifying it quite a lot, but at least we've got the narrative. So we've got an evolutionary survival threat response. We get a threat, we need to survive. It's based on our genes. It's very instinctual. So what's happening? We have a stressor. So maybe that's a scary line jumping out at us. That stressor is detected by hypothalamus. That's going to do two things. It's going to trigger the endocrine system through what was known as the hypothalamus pituitary adrenal axis, but just HPA axis, and that will release cortisol. Cortisol acts very slowly on the body. But it's also going to go through another route, which is a lot faster, which is a sympathetic nervous system, and that will release adrenaline, and then that will have a very, very quick response on the body. So that's my, in two sentences, how do we sum up what the fight or flight is? An overview of its evolutionary explanation, but then also what's exactly happening in the body in the fight or flight. And if we've summed that down quite well. Okay, so let's go for what else we need in this section, because it does go on to say we need to know the effects of adrenaline. So let's talk about what are the effects of adrenaline. Just out of interest, guys, everyone's freaking out about evaluations for fight or flight. Can anyone point out a single year in the last 7 years of this specification being live that we needed an evaluation for the fight or flight response? Because I keep a relatively good eye on it, and I've never seen an example. Oh, Stu, that's really kind of you, man. Thanks for giving me a 5. That's incredibly generous. But yeah, really, really nice for you to put that in the chat. If you're not already a patron, make a free account, drop me a message, and I'll make sure I'll put you in the Discord for tonight and the next couple of weeks before paper free. That's really kind of you. Don't want you to give me a 5 if I'm not getting anything out of it. Yeah. So yeah. Okay. Yeah, never say never. Like, yeah, the wise pleb, you're right. Never say never. But I mean, you can play this game. Like, your anxiety levels, everybody right now, is probably pretty high. I understand that because you don't know what's coming, and it's scary. But you can't let that completely take over. You've got to be relatively realistic with what you expect from yourself at this point in the game. You will not be able to answer every single question perfectly. I don't know how many people have watched my video about the grade bounds for the different levels. To get an A star, you're looking at 75%. That means out of every 4 points available, you're only getting 3. Now that's pretty wild, but I mean there is a reality that only 6% of students on average per year, that varies a little tiny bit, get that A star. So, a lot of people, a lot of the time, are leaving points on the table for a lot of reasons. So to over worry about things that haven't happened yet that could happen, sometimes isn't the best use of your mental resources. So yeah, don't freak out too much by what you read on the internet about people worrying about certain things. We can base most of what we know on previous years and what we've seen. And if a question comes up that people aren't prepared for, which kind of happens every year, the AQA might ask a question that's a little difficult, and we all adapt and change how we teach things on the basis of what we see in previous years. Don't worry. It's likely that basically every other student across the country has opened that page and gone, oh my gosh, how am I going to answer this? And had a really difficult moment. I really do think, rather than losing points for the majority of you on things that you haven't revised or didn't memorize, far, far more of you are going to lose points by not reading questions properly. You'll have the right information, but you won't shape it to the question asked. And, you see this time and time and time again as a classroom teacher, not shaping answers to the question asked. So, yeah, chill out. Okay. Yeah, I think I've only ever had one student that got 100% on a paper. Gee, that's not quite true. Yeah, I'll across 2 papers. Okay. But it's not necessary. Yeah, some people are amazing. Okay, so what have we got here? I know a bunch of I've all typed this. Sorry, I didn't pick up on it because I was talking to you through that freak out that most people are going through. So it can ask you about the effects of adrenaline. And then we want to go through the different elements. What's happening when we have adrenaline? Our anxiety is increasing, our attention is increasing, and our alertness is increasing. They're all psychological effects. If you need to talk about psychology, we're thinking about behaviors, we're talking about mental processes. We're not just talking about like physiologically what's happening. So if you're thinking about psychological effects, we feel more anxiety, we're paying closer attention, and we are just generally alert. So paying attention to particular things, but we're generally more alert. There are physical changes. So we have blood flow to the brain and muscles, because obviously we need that to think quickly. and reactions, dilated pupils, so obviously you can see better, breathing rate down, blood flow to skin, and digestive and immune systems. Yeah, I've been a psych teacher in the classroom for almost 10 years now, so yeah, been a while. So reduced flow to the skin, you might wonder, why is that? Why are we getting reduced flow to the skin? Well, we need it for our muscles more than our skin. And if we get cut, we don't want to bleed out. So yeah, reduced. Okay. Let's see. I don't think I have any other questions in there that I need to answer on that. We could go into the pack, but I'm not really sure on the question. Maladaptive to the demands of the modern world. Obviously, we got this as an evolutionary threat response. The things that make us scared these days, like psychology exams, can't be run away from, really. And they can't be dealt with properly at the fight or flight response. If you're feeling the fight or flight response now for the psych exam, I can understand why, but it's not helpful to us. Especially that, not so much the adrenaline, but the cortisol just builds up over time and does a number on your body systems. Okay. We are so maladaptive means working against your best interests. So it might have developed with a good reason, but now it works against our best interests. So yeah. Okay. They're not necessarily always 616 markers. I think there was 1 year where there wasn't, but there generally is overall. I could be wrong though. Okay, we are moving on now to a big section: localization of function of the brain. I'm going to try and keep this snappy. I know we're 51 minutes in. We've done the short sections now. We've got quite a few long sections coming up, and I do know you've got a lot of other revision. So I will try and keep this going. If you have questions in the chat from previous sections, hopefully other people in the chat will pick up on them and run. But let's go with localization of function in the brain. Please, someone just give me a definition. What is localization of function in the brain all about? Okay, Lola getting in there nice and quick. Thank you for that. Specialized function for each spot of the brain. So Broca's areas of speech production. So areas of brains are responsible for particular functions. Yeah. Okay, we've got some really good ones. Specific functions are found in particular areas of the brain. Brain hemispheres from Chris. Okay, some good examples of sections. There are parts that we need to know. We can ask about directly, and I've got those labeled up, and we'll work through them. We just pull up, because I do have the spec here on the page. Yeah, so location of function of the brain: hemispheric, lateralization, motor, somatosensory, visual, auditory, and language centers, Broca and Wernicke's area. So lots of like individual sections here that they can really like drill into. So keep in mind, I've given you for each of these its function and also what happens when it's damaged as well. A lot of you doing great work already, just popping in the chat what the different labels are. I'll go through those in a second. So next up, what I want to do, although I'll reveal that, localization function of the brain. So we've got some functions. So it's just the fundamental argument that functions of the brain, like speech, language, memory, are performed in distinct areas, not holistically. Memory is an interesting one, isn't it? Because the processing of memory might happen in a distinct area, but the idea of an engram, particular memory being located in one place, doesn't seem so logical or accurate. So it's not saying that the brain works as a holistic net and all of the brain is equally responsible for each task. That's our distinction, and we can be pretty sure there is localization of function based on the research that's behind it. So the next thing that I want you to define for me is what is hemispheric lateralization, guys? what does the term hemispheric lateralization mean? And I think I'm going to answer gold prism in this response, so I hope that's helpful. Merida's two halves of the brain are functionally different. Yeah, the corpus callosum like enables discussion between them. We're not getting split brain just yet. So to say that hemispheric lateralization, each hemisphere is specialized. So the language centers are in the left, and the visuospatial tasks are best performed, not exclusively, but best performed by the right. So we actually have a distinction between the hemispheres. So obviously, if you get the hemispheres, if you get your brain out of your skull, we'd have it about that size, the size of two fists, and then it would open up like that. Like a... Well, actually, if you picked up your brain, it would probably go through your fingers because it's the consistency of custard. But, you fix it in some chemicals and you can hold it, and then you'd see it like that. So there's the two hemispheres. Let's go now into defining some of the sections. So give me A. What is A, and what's it for? Okay, well, I'm going to define contralateral a few times. Some people asking for it already. So this is the idea that one side of the brain is responsible for the sensations or activity on the opposite side of the body. So yeah, the motor cortex here is the correct answer. It is contralateral, which means that the right side of the brain will control the movements of the left side of the body. And you could, like, if you an interesting element of this is, like, if someone was having a stroke, you could actually, and then they lost like their ability to move their arm, you actually know where the stroke is from that point based on which arm fell, and you would also be able to identify it even closer because there's a particular part in the motor cortex which controls the arm. So that's contralateral, and I'll talk about this a few times. Yeah, motor area and cortex, same thing. Yep, but I'd go cortex. Because the cortex means, I think most of you know what cortex is, but maybe not. It's the outer layer of the brain, whereas the complex processing isn't deep down. The deeper down, you have got white matter. It's white because of the myelination. The processing is happening on the surface, and that's why it's so like bobbly. I don't want to say bobbly, wavy. All the folds to increase the surface area. So yeah, about like an orange peel. Can I think about strokes we're using A3 for evaluation? Maybe, but I've got better evaluations possibly. Or you said maybe the knowledge of location of function can be helped in medical situations. I'll take that. Okay, let me reveal that. Motor cortex, voluntary muscle movements, contralateral. Let's go into a little bit of extra conversation on this section. What's going to happen if it's damaged? Well, on the opposite side, we're going to have, if it's a mild bit of damage, we'll have muscle weakness. We'll still be able to use it, but we'll have muscle weakness. Severe damage would be paralysis. So yeah, depends on the level of damage. The next one, everyone, B, please. B. Okay, yeah, loads you go into somatosensory cortex. Well done. And let's talk about what it's for. Sensations. A lot of people talking about sensations. What do we mean by sensations? Lola going in really well with somatosensory cortex. Receive sensory impressions. Damage results in loss of sensation on contractile side of the body. That's a nice little definition, including damage. Yeah, we've got heat and we've got touch and Kitty in there with pain as well. I'm not sure if someone else mentioned pain a little earlier on. But there we go. Somatosensory cortex receives sensations, touch, proprioception. Proprioception is like you close your eyes and you kind of like know where your hand is, like in 3D space. Like that's, we've got a lot of senses, and that's one that's not really talked about much. We kind of know where it is, and that's proprioception. And obviously the opposite side of the body, it's contralateral, and damage is loss of sensation, numbness. Okay, moving on to C. What is C, everyone? Yeah. George in there first with Broca. Rin with a lower definition, controls speech production. Damage leads from Broca's aphasia with speech is impaired. Yeah, Broca. Okay. There's a little bit more detail. So Broca's area, left frontal lobe only. Interestingly, I don't know if you guys know the percentages of this. It's 90% or 95% of right-handers have got the Broca's area in the left frontal lobe, and 70% of left-handers do as well. So the vast majority of people do, but it's not actually. Hey, Sass. Hi, Tutu. Did I put two Ds on the diagram? Oh, well, I will fix that and then re-upload the digital version. Thanks for that spot. Left hemisphere only, mostly. So yeah, for speech production, yeah, obviously Tan is in there. And we lots of different aphasias. I used to use motor aphasia for this, and then I did a bit of research recently and say that it was the original term for this. Obviously, you can use Broca's aphasia as well, but it just seems like a bit of a cop out, doesn't it, to say Broca's aphasia for Broca's area? But you can do it. So now apparently the correct terminology is expressive aphasia. So yeah, and its damage would be expressive aphasia would just be difficulty producing fluent speech. It doesn't have to be as extreme as Tan. You can have expressive aphasia and start to struggle over just being fluent in what you're saying. Cool. And then let's go for the purple D arrow. Let's go purple, not red, so let's go purple. Yeah, okay. So in there we have Wernicke's area. Jen has gone Wernicke's area, speech comprehension. And we've got Wernicke's aphasia. We can improve speech, but unable to understand language. So you guys can like word salad, don't you? Almost making weird noises. Yeah. Okay, so Wernicke's area, left temporal lobe only, speech comprehension, damage equals, we've had this before, so someone mentioned it, sorry I didn't pick up on it. It was receptive aphasia, difficulty understanding speech. Okay, give me the red area then, please, everyone. What's the red area? We got Zoe straight in with auditory cortex. Auditory cortex. Yeah. Auditory sound, received and processed audio info. Yeah. And where's this found then? Where's this found? What's different about the auditory cortex and Broca's and Wernicke's? Yeah, it's in the temporal lobe. That's true. Wernicke's in the temporal lobe as well. Avid first one in there. Yeah, it's found on both sides. So unlike the language centers, the auditory center is in both hemispheres. So yeah, contralateral still. So we're dealing with sound coming from opposite ears, receiving and processing sound information. Damage. If you have damage on one side, it's not the end of the world because obviously you've still got an entirely functional auditory center. So, but what we have, we're quite good at there's like a noise moving around, we can identify the location of it with the two auditory centers. Maybe a bit more difficult with only one. But yeah, if we have damage to both, which is quite rare because they're on opposite sides of the head, that have to be pretty severe brain damage for that to happen. Then we can get cortical deafness. Yeah, it's in both hemispheres. They're not. No, Broca's and vertigo aren't in the auditory cortex. The arrows and the locations are. You think about like language is a relatively like new thing that we have been able to do as creatures. Like creatures have had hearing and sound processing for a very, very long time. So I kind of feel like the language sensors have there are more recent development and taken new areas. So yeah. Yeah, I noticed there's 2Ds. I'll fix that when I re-upload the digital file. But if people have already downloaded the digital file, I'm sure it'll be fine. Okay. I think I have said enough for that. We're going to finally do the E. What is E, everybody? Yeah, it's frontal lobe motor cortex. Sorry, I didn't pick on that. Okay, we've got occipital lobe, but we've got the visual cortex as well, which is very similar. So when we have visual cortex, which is the occipital in the occipital lobe, and we've got both sides, and I think people are aware from the split brain stuff, if you know it quite well, that it deals with not separate eyes. The trick is to go as not separate eyes that it's dealing with. It's still contralateral, but it's dealing with visual fields. So that visual field, my left visual field, is dealt with the right lobe of the visual cortex. My right visual field is dealt with by the left. So yeah, just a little thing that might be problematic. Yeah, I've obviously messed up the letter, and then I'll fix that later. Let's do a couple of evaluations then. How did we know about Broca's and Wernicke's area, and how were many of these locations initially discovered, found, researched? Cheers, Ace. Yeah, post-mortem, Tan. People are getting excited about Tan. Yeah, you know, Tan, kind of sad that he's like Tan is named as Tan, because it's like, it wasn't his name. Like Groot's name's Groot. I know Groot can only say Groot, but like Tan's name wasn't Tan. It was Louis Le Bonnier, very French name because he was French. And yeah, like, and it's not Louis's area, it's Broca's area, which seems a little mean, but oh well. Phineas Gage, interestingly, yeah, like stuff about the frontal cortex, maybe. Yeah, it's Pokemon. Yeah, it's love, isn't it? Yeah, like calling the guy by the only words that they can say does seem mean. But yeah, case studies is how we originally found it. Can people criticize the use of case studies for localization and function research for me? So, historically, yeah, it's been useful for us to, and it's not just like Tan, like there's lots and lots and lots and lots of clinical case studies that show us areas of the brain, and they're often quite similar. But yeah, we do have that lack of generalizability. Why? I'm just going to push you a little bit on that. Why about the lack of generalizability? Yeah, Joshua there coming in with a really good addition there. They could have other brain areas damaged, and you can't study a person before injury. So we don't have it before and after because we know it's going to happen. And yeah, we can see like with the post-mortem, we can see the large damage, but there might be additional areas of the brain damage, or the damage might be more extensive than we think. So we can't be very, very precise when it comes to case study. But we can't not like be impressed by the amount of case study historically that's come about and pick up on that's where we've found most of these things. So large numbers of clinical case studies demonstrate a loss of functions, damage to particular brain location, like Broca's and Wernicke's case studies, aphasia and Clive-Waring amnesia, severe damage to the hippocampus. Actually, I'll tell you, like, how we know about a lot of this stuff and actually the details of the somatosensory cortex and motor cortex. I don't know the name of this surgeon now. It's a shame. One of you might do. There is a surgeon that worked in. Oh, Amelia, that's really, really kind, Amelia. You're very, very welcome. Again, like the other person, drop me a message on, make a free account on Patreon and send me a message and I'll pop you in my Discord. You don't need to do it that way, but that's kind of you. So, yeah. Anyone know the name of this researcher? The surgeon is a Canadian. Over a course of a very, very long surgical history, he was a brain surgeon. People will come in with tumours, but what he would do was he'd be like, well, I've got your head open anyway. Like, I've already removed part of your skull. Can I have a little poke around with an electrode and see what happens? And people agreed, They're like, well, you don't want to annoy the person who's doing brain surgery on you, so maybe there's a little bit of, I don't know, maybe it's a little problematic. But they poked around with the electrode, and what would happen is hands would start moving. Oh, Penfield, thanks, Farah. Thanks, Kara. Oh no, wait, that's the one. That's not the rat one, is it? Where they take part of the rat's brain? Well, maybe it's Penfield. Probably Penfield. Wilder Penfield. Okay. So yeah, start poking the brain with electrodes. Things are moving. Smells are coming out. So yeah, a lot of it was done through that, which was a very impressive way of just I'm going to find out about the brain because I've got these brains open anyway. So yeah, historical was great, but unscientific as damage covers multiple areas. So how do we do it now, everybody? Oh, Lashley was rats. Yes, good. Yeah, Lashley. Lashley's a counter. I've not included that. I didn't want to give that much detail because I thought we had enough evaluations here anyway of what I provided. But some of you are aware of Lashley. I'll just go through it quickly just because you're going to hear me now and you might want to say it, want to hear it. So you get rats that run a maze, and Lashley was the opposite of location of function. They were like, okay, well, let's just remove parts of these rats' brains. Gross. Not enough to kill them, but let's just start taking away random parts of the cortex and then let them run the maze. And what Lashley was trying to do is he was trying to research the concept of an engram. So the idea of an engram is that we have a memory and it's stored in one particular place. So Lashley was like, if I can find the bit where the engram from the maze is, I'll just take this one part out and then the rat won't be able to run the maze anymore because it'll have forgotten how to do it after it had been trained, but was never able to find the part of the brain where the engram was. Just basically how much of the brain was taken away, the worse the rat would do, but it didn't really seem to matter too much which parts. So yeah, that was a Lashley study. Suggests that the brain was more holistic. Okay. Yeah, that's just it's a bit grim, that one. People talking about Torvings. Yeah, go with Torving. Torving is a good one as well. Like, you're at this point now where you can probably see the links between lots of different psychology sections, and you can bring other areas in. But if you're confident, okay, let's go. So what we've got now is we've got modern fMRI studies that support case studies using healthy brains. And we are criticism, and you could maybe bring in Lashley here. There are many brain functions that appear not to be localized, like consciousness certainly doesn't seem to be localized in one particular part, but also memory storage. So I talked about Lashley then, the idea of this engram doesn't seem to be located in one particular place. Memory processing certainly seems to be. Obviously, we've got Clive Waring. His amnesia was due to the hippocampus. Oh, Yusuf, that's really kind of you. I'm not expecting anyone to give me donations today, so just doing this for funsies. But yeah, thanks for that. I also am not running ads on this video because I imagine it would be so stressful for it to cut out when I'm mid conversation and to run an ad. So yeah, that's kind of you. I think we're good. I'm done here with localization of function in the brain. I think hopefully you guys are happy with what we've gone through. Just trying to think whether I want to do another section before a break. What are we on? 114. Do people want breaks, or do we just want me to try and like go through and okay. I'll see the first like 20 people. You guys are not making this easy. Can I do a poll? Can I do a poll here? Oh, I can do a poll. This is my first time doing this, guys. So, Uh-oh, what just happened? Are people seeing this? You guys are incredibly evenly split on whether to be a break or not. It's relatively stressful. Okay. We have a very slight swing towards yes for a break. So we'll take a break, but I will maybe not have a very long break as a compromise then. Should we do that? Okay, so I've edited the break out of this video. Just a quick reminder that if you've made it this far, to subscribe to my channel. It would mean a lot, especially being so close to 100k. And if you want the digital version of this poster, please grab it on my website or the entire revision book on Amazon. Let's get back to revision. Hey guys. Mip. I want to say hi to my cat. Boop, boop. He had a little starring moment in my approaches videos. He hangs out with me most days when I'm making videos. But yeah, everyone would appreciate a cat break. Get off your nose. Yeah, his name is Obi. He's so greedy. Go that way. What's his opinion of the psychodynamic approach? He isn't a fan, you know. He's not a fan of the much. No, Obi, not Toby. Although, yeah, kind of sounds a lot similar. Like Obi-Wan Kenobi. I mean, I wish I'd look like Obi. Right. I got to get back to this, mate. And I know you're going to be angry I didn't give you that whole thing, but you have to go. Right. Yeah. Yeah, I mean, like, my cat is a pretty good example of cupboard love. Sometimes I think he wants my attention, and then I realize he just wants a treat. Mate, you're going to go. You have to go. Bye. All right. Yeah. Next one. He can do issues and debates. Okay. Oh, say hi to Obi, your dog. Right. Are we want to get back? My concurrent viewers went up quite a lot then for I put the cat on and not me. I don't know what I was meant to say about me. I bet I'll go down now because like Obi's not on the picture. Yeah, concurrent views 323. Nice. Good to see you all. I know that a lot of you won't be here from the start of the stream. Shout out if you've been here from the start of the stream though. I know it's a lot to get through these few hours of revision, especially in this last day before. I know it's quite stressful. So just a quick wise pleb. Yeah, I remember from the beginning of Kitty. Yeah. Well done for sticking around for the full 2 hours or so. Just a reminder, these are all live streams. I'm on my 6th one now. The approaches one has disappeared. So if after this you want to do the approaches one, you would have to be a patron. And you want to access the posters, including the comparison one as well. Although I did put them up on my website for like £2 each, and they've done pretty well. And if you are a patron, I would recommend doing some of the research methods past papers, because that's probably a good way to finish off your revision with a massive research methods section. But I won't say any more on that because I will get right back into it. Examine 18 hours. Wow, I wonder how many times you could watch my like in 20 minutes in 18 hours. It's pretty a lot. Um, especially if you did on 2 times speed as well. I don't know how many people watch me in 2 times speed. I once had a student who watches me quite a lot. Well, I didn't teach him in second year. And then I spoke to them and they were like, oh, it's so weird hearing you speak in a normal speed because I just listen to your videos at 2 times speed. I'm like, oh, okay. All right, let's go into this. I don't think the other half will take as long as the first half did. I imagine bioribbons will take a little while, which is annoying because obviously that's coming next year. But let's get into plasticity. Yeah, I feel like 175 is probably the sweet spot for speed. 2 speed is a lot. Yeah, I'm gonna do split brain research. Definitely. We've got a lot of terminology here in plasticity and functional recovery. A lot of what we do here is going to be based on like, do we know this term, that kind of stuff. So, first of all, give me plasticity. Just what is plasticity? So much to me a 0.75. I speak slowly, I think. So I'm going to, yeah. So I'm straight up with Maguire. We're not our evaluations yet, but like the enthusiasm. Yeah, the brain changing to adapt is good. Great new neural pathways associated with learning from Grace. That's good. In response to learning or trauma from Adam. That's good. So I know a lot of you are saying the same thing. I'm not picking up on all of you, but you're all doing great. Harry Maguire study. Was Harry the first name of the person called Maguire? I did not know that. Okay. So, I've revealed too much there. What is functional recovery as well? Because I'm going to reveal 2 terms. Plasticity, I've got the brain adapts and functions and structure due to a change in the environment, which could mean damage or learning. What? Okay, functional recovery there, guys. What is functional recovery? Yeah, Lisa Mellon there with a really good short definition. Healthy area is taking control over the areas damaged. So it's not necessarily the brain area recovering. It's the task itself, it's the function that is being recovered. But yeah, Celeste there with a more detailed one. Functional recovery is the brain's ability to recover lost functions by having undamaged areas of the brain take on the role of damaged areas. And I will have an example of that for you in a second. So yeah, plasticity, the brain adapts in function and structure due to change in the environment. Damage or learning is included in that. Functional recovery. Undamaged areas take over functions that used to be performed by damaged areas. I know everyone typing in the chat right now is a subscriber. But yeah, I'm getting close to 90k. So if you're like just watching me and not chatting, then yeah, subscribe. That'd be really nice. I'm not getting to 90 this year, but it'd be nice to get there by September. Let's go for another term, which is synaptic pruning. What is synaptic pruning? Thanks, Fatu. I'll get there one day. So ends in there with removal of all connections, destroying unused neural pathways, efficient synaptic connections get strengthened as well. An element really of it is like, look at the contrast. Charlie, SAMs that are frequently used become stronger over time. Unused connections are lost, more efficient. That's good. Yeah, Jake, isn't it weird that babies have the most synaptic connections? I don't know if any of you guys have watched a documentary series called The Brain by David Eagleman, but if you're into this kind of stuff, it's like the best documentary series ever. They have a good explanation of that. Loads of good stuff for the spec in there. Okay. I would like another definition. I know I'm good. Synaptic pruning synapses that are used frequently get stronger. Unused synaptic collections are lost. Synaptic pruning is for plasticity, right? Yes, it is, because it's about the idea of being plastic. Like the word plastic for you guys probably means like like a hard plastic substance, but plastic means moldable. This was the name of it. So it's the brain molding itself to the demands of the world around it. So any physical change, that is basically then leading to functional change. But with like, how is the brain actually moldable? Okay. Next one I want then is a bit of a fancy word. Don't worry if you haven't heard of this, because I think we've got enough with what else we said to get what we need. But does anyone know what denervation supersensitivity is? What is denervation supersensitivity? Dopa just like, no. Okay. It was in a mask a while ago. It's something that you could use in this area. It's absolutely fine. Leon, though, with a definition as well, right? Freya also right. Celeste also. It's like, if you say, is this on the spec? Like, there are lots. All the spec says, and I'm reading it now: plasticity and functional recovery of the brain after trauma. So that's what's on the spec. What you need to know around that concept, it's kind of up to you. Like plasticity, you need to define it. Functional recovery, you have to define it. Basically, all of you knew what synaptic pruning was. Synaptic pruning itself isn't on the spec, but it's an element of plasticity. In the same way, denervation supersensitivity is one, and I think it's been in a mark scheme, possibly two, but at least a mark scheme. So a few of you kind of had this right. Let me unveil it for you. If you've got like a pathway of axons, like a large number of axons that all are in the same pathway, I guess. If we have some of them lost, then those remaining axons will become more sensitive, so that we are still able to produce the function. It's in some textbooks as well. It can cause pain as well, because they're potentially too sensitive now. So it's problematic, but it's it's a kind of emergency situation. All right, I think that will be enough to talk about plasticity and function. All right, then, you've been wanting to do this for a while. Give me the Maguire study. Like, first thing, everyone's going, Maguire. So we've got taxi drivers. What else are you telling me? What else? Posterior hippocampus is what we're looking at there for 9 PKE 21. Yeah, larger than control group. In London. Higher volumes of grey matter in posterior hippocampus. A little bit more detail than I went into. Okay. Yeah, some extra little findings there. The longer they were, the bigger it was. Actually, I didn't mention as well factors affecting recovery. Children and women recover better. We could pop that in. But the Maguire study. Oh no, I've got 2 studies there. Just a second. All right, I'm back. It wasn't that much of a technical issue. I just wanted to reveal them one at a time. So we have got MRIs, 16 taxi drivers who took the knowledge. So that, as you're aware, that's the test that all taxi drivers have to take in London. An incredibly detailed memory challenge, and then obviously they'd been working taxi drivers in London as well. So that massive challenge seems to be correlated, because there was correlation, with significantly larger posterior hippocampus than the control group, suggesting that the brain will physically change, not just to trauma, but to new learning as well. I assumed you might like seeing the technical difficulties slide. May you still filling these in yourself, like with a pen and stuff? Okay. The next one I want is a study, if you know it, by Daniele, and we're going to use this to talk about functional recovery. That's a new one for me. Boy taught 60-year-olds to juggle. Great matter increase of visual cortex effect removed from practice stopped. Oh, that's interesting. I did not know that one. You could use Lashley again. I don't know, because I feel like Lashley was like they cut open the rat's brain and the rats were just made to do their thing and see where they were without that part of their brain. Like plasticity and functional recovery implies that there's enough time for the functions to return, which I don't know if the rats live that long. Oh, maybe if you could send me some pictures of those completed posters, it'd be great. I've never seen anyone else do one of my posters, like how it's been filled out. So, okay, yeah, EB case study is what I'm going for. This is wild. And if you go on YouTube, you can see interviews with a kid. I don't know if it's the same kid who had had half their brain removed. I mean, it's absolutely wild, like how much of your brain you can live without. Obviously, if you're an infant. And then still kind of like exist normally. Like it makes, yeah, it's very odd because we just assume that we've got all this brain and we need all of it, and we kind of do. But then you have these situations like this, and there's another one as well where they scanned someone's brain and it was basically all cerebrospinal fluid. There was very little white matter in there, and it was almost just like just a cortex and like a very small amount, and they seem to be okay. And then there's Mike the Headless Chicken, which you probably don't know about. Like there's this chicken and actually maybe I shouldn't talk about Mike. Okay. So got 2 years old, EB of the left side hemispherectomy, removing Broca and Wernicke's area. So yeah, we took the left side out. They had some basic language at that point. It all disappeared. And then EB over 2 years recovered that language ability. So yeah. A good example there of functional recovery. We lost a function and then we regained it. Who was a 14-year-old? So I didn't want to put in like, oh, they were 14 when they like did the study. But like what's more important, I think, for this section is they were 2 years old when the hemisphere was removed. Oh, no worries, man. So yeah, I mean, yeah, if the kid was 14, it wouldn't have worked. They would have just died. It's the ability of very young children to have significant recovery from brain trauma is very impressive. But yeah. Yeah, I'm not going to tell like Mike isn't an evaluation. Mike isn't a anything. It was just kind of interesting. Like in the olden days, there was like a traveling circus, and they had a chicken who'd had its by the farmer had gone to take its head off, but the chicken still lived without its head. But that's because there was enough of its brainstem around for it to do its basic functions. So it was pretty wild. There's pictures online. So yeah. Okay, we have why was it removed? It was a tumor. There was a severe tumor. The kid would have died without the tumor removal, and basically the tumor taken over the entire hemisphere anyway. So yeah, just there was no choice, and it worked, which is great. If it is the kid in the video and they're like play football and stuff, absolutely wild. I think we could use both. So Daniele is the question. So the section it is, the bullet point says plasticity and functional recovery of the brain after trauma. So these are doing separate things, aren't they? Daniele is looking at trauma, the removal of a hemisphere, and then we have recovered. So we've got back language ability, which was lost. So that's functional recovery after trauma. Maguire is doing plasticity. So we've got an adaptation to the environment. The environment for these taxi drivers, they had to do a lot of learning, and the brain physically changed in order to allow that learning to happen. So yeah, there is a crossover certainly, but there is definitely two different things that we're going with here, and that would make a nice little evaluation section. Yeah, you could incorporate functional recovery in AO1, definitely. Okay. I don't use Genie for anything, really. Okay, and the next question I'm going to ask you is, can you tell me about a practical application? I've got a nice term for you. Hopefully some of you will have it, but don't worry if you don't. There's actually a pretty good practical application for plasticity, understanding it. So we've got neurorehab. Neurorehab, what's the thing? Oh, there we go. X_AL, straight in there. I wonder if the rest of you know about constraint-induced therapy. Spike does as well. Yeah, we can talk about functional recovery if they talk about plasticity, because I guess functional recovery is an element of plasticity. Straight moving, but so yeah, a bunch of you know this. So we've got practical applications through the straight-induced therapy. So stopping clients using coping strategies makes them improve through functional reorganization. So basically what this means is maybe if someone loses the ability through neurological damage to move their arm well, they'll tend to not use it. But what could happen is if you tie up their good arm and force them to use the bad arm, that'll make the functional recovery faster or happen at all, because the brain is kind of like forced to make those choices. Next up, we have a criticism. Can anyone give some criticisms? I've got one. I reckon there's probably quite a few criticisms that I've not included. So let's see what we've got in the chat as well. So like eye patches as a kid, yeah, it'd work. Yeah, I've got the Mafia evaluation as well. Sample sizes and studies, problems with case studies. Yeah, we can criticize the Daniele one for that, but wow, like it's hard to argue against somebody who's had half a brain removed. Yeah, it's hard to talk about like criticisms of plasticity because it kind of like it kind of is what it is. I mean, a lot of the biological stuff is so well evidenced. It's a little hard to argue against. It's not like there's not much subjectivity here. Like as I said, like I mean, even like criticizing case studies and being like, oh, it's not very generalizable. Like it's not, but I mean, they definitely had half the brain chopped out, and they definitely survived, and they definitely regained language. So it does suggest that it's possible. It's not possible because this person definitely had it done, and it's happened multiple times. But we have to do what we can with what we've got. We do want to evaluate. We do want to potentially have criticism, and yeah, we were going to go for this one. So Mafia's meta-analysis, basically, plasticity recovery is a thing. We can go with that, but not everyone is really able to have plasticity and functional recovery. So there's even though these concepts are out there, maybe there's a lot of variability. So Mafia, not everyone has high level of plasticity. Some participants have high levels of cognitive reserve. So that's the variable that we're looking at, which is saying just plasticity on its own isn't enough to explain. So intelligence seems to be a large factor, and also level of education as well. Yeah, educated students do better. So yeah, I guess this is the one that everybody else goes for as well. Okay. Let's move on, because I do aware we're almost 2 hours in. Although we don't have many sections left, you're doing well. We are going into split brain, everybody. Split brain. We have 2 sections after this. So first of all, can you just tell me what contralateral is, please? I know we've talked about this before, but let's go contralateral. Yeah, this is a good one. This one's like, of the bio videos, the one that seems to get watched the most, and I think it's because random people are watching it. Sperry, yeah. Opposite side of body controlled by hemisphere. And what is cut and why? So it's a split brain. What was cut and why? Yes, by coming to a corpus callosum. And Rosie ended with the idea that it was epilepsy. CC talked about reduced seizures. A lot of you coming in with the right answer as well. So our setup is the argument. The brain is contralateral. Each hemisphere controls the opposite side of the body. That's important with how the study was run. The hemispheres are connected by the corpus callosum, which is 200 to 300 million nerve fibers, which allow the 2 sides of the brain to communicate. And cutting this for the treatment of epilepsy, I kind of feel like I needed to have said that before actually mentioning Sperry at all. So, what did Sperry do? Don't tell me about the Sperry's findings just yet. Like, what did he do? Grace pre-guessing what I'm going to say, and did quite well. Using the very fancy word tachyoscope, which I've not included, but is true. A little device to control where people looked, or what people could see. Okay, yeah, use field splitter. Nice. Show them images of both the visual fields, get them to talk about it. And they've got JJ to say, draw, or pick up what they saw. Good. I think you've done enough there to pull together the setup. So 11 split brain patients. I know that we mentioned there was a control group of 11 as well, but the actual split brain patients were 11. They'd all had a corpus callosotomy of some level. So we show a different image to each visual field. And some of you talking about the device that was used. But fundamentally, we show a different image to each visual field. And I've done a little diagram there of what it means to have different visual fields come into each eye. And then that information was sent to the opposite hemisphere. And then they were asked to say what they'd seen, or to draw, or to select what they'd seen. What was the findings, everyone? What was the findings? Yeah, patients could not verbalize the stimuli presented in the left visual field as they were processed in the right. Left hemisphere could be spoken from Lola. Right hemisphere could draw. Okay. I think we're good. It's a complicated little study, so you've done well to summarize it in a sentence or two for most of you. So Sperry found info presented to the left hemisphere could be spoken, location of speech center, the right way around. Suggest both hemispheres are capable of independent awareness and action. Additionally, language centers in the left hemisphere. Yeah. What have I done? Yeah, that's fine. Okay. That's what you guys put in. Very good. Is it all right if I only talk about the speaking as part of the study? Yeah. I guess it is okay if you do the speaking part of the study, because obviously one side couldn't be spoken. But I guess it's kind of interesting that the hand could pick up the hand could pick up the object on the opposite side, because it kind of means that it, like, there was another consciousness. The fundamental element was like, it seems like there's 2 people there. like two distinct parts, two distinct identities. So it is interesting that the other side could still do something. I wouldn't go into too much detail here. I think this is probably enough. I just wanted to make the distinction between like being able to select an object and the right hemisphere being able to do that with the left hand. But then I guess the fundamental one is about being able to speak, because shows both hemispheres are capable of independent awareness and action, but additionally the language centers are in the left hemisphere. Does anyone know of a different split brain piece of research? Because it doesn't say in the spec Sperry split brain, does it? It just a split brain research. What's the other study? Yeah, Gazinga. Okay, what did Gazinga do? What did they find? Okay, yeah, facial recognition. So we've got something for the left hemisphere, but we found something for the right hemisphere as well. Split brain research. Each hemisphere was showing faces. I struggled to get into a sentence, but each hemisphere was showing faces. And what we found is the right hemisphere is specialized with facial recognition because it was better at it. So at least we've got an element there for left and right. Do we need to include both in an essay? Probably not. But if you've got an essay and you know it, then go for it. Let's go for some evaluations, everyone. I'm pretty sure I know what evaluation we're going to go for, but let's throw it in a critical evaluation. What's the problem? So Rin criticism control group didn't have epilepsy, so it's not clear if it was linked to epilepsy or cutting the corpus callosum. Yeah, I think a lot of people going down that way. The medication as well. So yeah, the Kira's got that. Kira, the medication and severity of the corpus callosum of cutting it varied between them. They weren't like necessarily all severed completely. So yeah, confounding variables. A lot of people putting in. I think we maybe also mentioned, I may see there, but there's probably some others as well. The small sample size is problematic. So I think we're good for critical evaluations. Has anyone got a good one for me? I mean, it's a good study, really, but it's quite easy to criticize. I mean, we could maybe talk about how it supports the idea of language sense in the left hemisphere when we kind of basic, but it's pretty well controlled, standardized procedures. Yeah, no ethical issues because they had already had the corpus callosum severed. Okay, so not putting the one I've got, I don't think so for the good one. I think it's quite impressive in this way. So I actually first learned about split brain research when I was doing my philosophy and psychology degree from the philosophy side, because it is very well known outside of psychology, because of this indication that there is more to the mind than a single consciousness. We see here, an ability for one mind or one brain to hold multiple minds. So that brings big questions about consciousness and big questions about identity, if they can act so independently. But yeah, so other good points in there as well from you guys about the control over the study. Let's go in then. We'll be relatively quick through this one. I think ways of studying the brain. So please, everyone, tell me what fMRI is. You can tell me what the letter stands for if you want. But I would like what is it? What's it doing, and how's it working? So L shows blood flow. That's good. Look at a little bit more detail. Magnetic analysis of oxygen in the brain. That's great. I didn't quite get the space to fill all that in, but you are right. But yeah, it is a functional magnetic resonance imagery. So yeah, the magnetic element is in there. High spatial resolution. Got some evaluations there. Let's say those for a quick second. We're good. fMRI detects blood flow in the brain. As more active areas need more blood, they can compare to low activation areas when participants are doing tasks. And you can mention this, create kind of a heat map of activation in the brain. And you've probably seen these before with like red hot spots and blue cold spots. So we get them to do a task. We measure their brain waves. I just realized I'm not very close to my microphone. Hopefully I'm being loud enough. So yeah, let's go on to evaluations. A few of you throwing in there. Poliski Vamper, non-invasive. Yeah. Lisa Mel, low temporal, high spatial. High spatial. Non-invasive is good as well. Okay. Yeah, a lot of the positive and negatives that I've got. So good spatial resolution, so down to about 1 millimeter, not ridiculously good spatial resolution. We've got another one that's maybe a lot better, but I mean, at least it's non-invasive, and also it's safe as well. It's not on the list, but there are some like PET scanners and stuff where you have you have to take a radioactive substance, so not useful really. So because it was temporal and spatial ability. So spatial means like physically how close, how far can you zoom in? Can you see like detail of the brain? So this is a good spatial resolution. The scanners are quite accurate, down to about 1 millimeter. But temporal resolution is like when you see the activity happening. Now it only takes an image every few seconds, and brain processes are very, very, very quick. Far faster than like every few seconds. So we're not really seeing exactly when it's happening, or the process of how the memory is working. So we can say it's got poor temporal resolution because it doesn't take enough photos fast enough. And okay, we're good. You can talk about Tulving and you can talk about Parapakaran, yeah, as it brings some things in from memory if you want as examples of fMRI studies that are good. I don't have space to put those in, but that would be useful. Let's go next for an EEG, everyone. What is an EEG? How do we do it? What's it getting? What's it finding? Okay, Olivia, ready there. Skull cap, around 22-34 electrodes. Yeah, we put those in the scalp, provide activation of brain cortex under the electrode, and we display the brain waves. That's pretty good. We got Luke T, measure electrical activity in the brain as a whole. Yeah, the measure activity in the brain under the electrode. Already potentially hinting at an evaluation there. Electrolitham gel from Arena. That's cool. Okay, we're good. So EEG, what is it? Displays brain waves on a cap, sum of activation under the electrode. That's going to lead us to our evaluations then. So Ellis has got low spatial resolution. Alfie says can't see deep activity in the brain. Arena's high temporal, poor spatial. So yeah, some of these match up down there. You can even have an evaluation in here, which I've not included, which is sometimes we might want to do some triangulation and do similar studies with different brain studying techniques and see the and combine them. Okay. And it's a second. Right. EEG evaluations then, please. What are EEG evaluations? Okay. Cheaper than fMRI from ACE. Okay, yeah, I'll just reveal these ones now. You've got there. Good temporal resolution. So we see obviously multiple, you see the feed out of these. They're like little arms with pens at the end, and they shake and then they create a brainwave pattern. We have temporal resolution in the milliseconds. So much better temporal resolution than fMRI, cheaper than fMRI. The problem is, yeah, we can't see deep in the brain, and yeah, we're just seeing under that electrode. We're not seeing general activity. All right, some of you are already talking about ERPs. Give me ERPs. What are they? I'm going to have to spend a little longer talking through this one because I know this one is particularly complex. Okay, really good use of that term of statistical averaging. I think that's something that we might need to talk about a little bit. Okay, we got some good answers there. I think there's one thing that a lot of you are missing out, though. Maybe not all, but most of you. Charlie's got it. Okay, the element that's important about this is with event-related potentials, yeah, we're using the same equipment as EEG, but we're presenting the stimulus not just once, but multiple times. So let's say you get someone to squeeze a tennis ball. Squeeze a tennis ball, and we keep doing that. What we would see is obviously you have like all these electrodes all over the brain, and some of the messages might be related to hearing the instruction squeeze the tennis ball and then squeezing it, but a lot of the brain activity is just doing random stuff like regulating breathing rate or thinking about what you had for lunch that morning, or just absolute random stuff that we have no idea what the activity is for. But the first time you squeeze the ball, it's going to be impossible to figure out, almost, it's going to be very difficult to find out what the brain activation was about squeezing the ball because of the random noise. But then the next time you do it, you'll still have random noise, but you'll have different random noise. But you'll still have the ball squeeze activation. And then we keep doing it and keep doing it and keep doing it. We'll always have random noise, but it's always different random noise. What you can then do is you can isolate the repetitive pattern that was used to squeeze the ball from the random noise. You're getting rid of all the random noise, and what you're left with is the activation that was associated with squeezing the ball or doing any task, any stimulus. Yeah, that's it's a tricky thing, but that's what we're doing is getting rid of all of the random noise the EEG produces from detecting all of the electrical signals under the electrodes. So yeah, hopefully that helps. But that's probably the most important thing about event-related potentials. So I will just reveal the evaluations for this. So we can isolate and study individual cognitive processes in a way that we can't with EEGs. So we're going to distinguish this from EEGs. It's that isolation and studying cognitive processes. But it shares the other strengths and weaknesses of the EEG. So again, good temporal resolution, but poor spatial, because it's the same kit, it's the same technique. And also, we got an issue that's, some processes can't be presented multiple times. Like if you're going to do like a fear response and shock someone, there's only so many times you can surprise someone. So we're not really going to be able to attest that with this process. Let's finally do postmortem techniques, postmortem examinations. What are postmortem examinations? What are we going to say? Yeah, I don't think we can say domain characteristics because you can't really control what your brainwaves are unless you're like a Buddhist monk, apparently. You definitely wouldn't be able to do domain characteristics on a post-mortem as well. Okay. Yeah, dissecting the brain to see any abnormalities. Broca's area, you've got an example of there. Gina's got a really good little chain there. You observe participants' behavior, examine the brain after death, correlate behaviors to lesions of the brain. Exactly what Paul Broca did, yeah. So let's give the A1 of just what it is. Post-mortem examinations, brains are precisely cut after treatment. So yeah, keep in mind if you take someone's brain out after they die, it's not going to be immediately workable. You'd have to treat it in a chemical substance to firm it up. But then you can slice it. There's like little brain knives, like bread knives, and you can do really nice, precise slices. And then what we've got unusual brains. So this might be people who had trauma or mental illnesses. And then you're comparing this to healthy brains. And yeah, as Mary says, correlation is going to be an issue there. And our great example there is Broca's area. Broca's area was identified by post-mortem. I'll fix that one. And then, yeah, let's just go. Actually, no, you can give me this. Evaluations, please, guys. What are the evaluations, positive, negatives for post-mortem? Yeah, we've got an unethical and, but yeah, I mean, I suppose these days you probably have to donate your body to science for this to happen. You might have a conversation with your doctor before you die. So I imagine most of the time these days we would get consent before death. Alex, overcomplicated one. Brain erosion due to death is criticism. Yeah, neurons break down pretty fast. Yeah, detail to the neuronal level is a positive. So we go low spatial. Now it's the opposite. Very, very, very high spatial. You can get like an electron microscope on these things and just really get down to a super low level. There's a really good I don't know where the video is, but what they've done now is incredible. They've got like the sharpest knife ever and a rat's brain, and all they do is they slide the rat's brain against this sharpest knife, and it creates like a strip, a really super thin strip of rat brain, and then they attach it to basically tape, and then they scan using a very high resolution magnifier the rat brain, and then they use the computer to rebuild the slices back up again into the rat's brain. I don't think they've done a whole one yet. I think they've done like they might have done a fly or something, and you can recreate the brain virtually in a computer, seeing how all of the individual neurons are connected to all the other individual neurons to just a ridiculous level of detail as a three-dimensional model. So yeah, there's some cool stuff they can do with this with good rat brains. Yeah, those rat brains are always so interesting. Okay, so we've got that's way too fast. High spatial resolution down to the microscopic brain structures neuronal level. But yeah, like all of our other ways of doing the brain, comparing them, remember, not on a living brain, so no activation. Damage in behavior then is correlational as well, because we're not necessarily doing the damage ourselves intentionally. Okay, we now, everyone, are on, and I'm sure you'll be happy to hear this, our final section. Yeah, final section, which is biological rhythms, which is a lot. So, very difficult sometimes to know how much detail to go in biological rhythms. Hopefully you'll agree I've made the right call on this and may have to summarize it quite well. So, please tell me just what a circadian rhythm is, please. What is a circadian rhythm? You got Olivia there saying the circadian phase ultradian. Yeah, we'll get through that. 24-hour cycle influenced by endogenous and exogenous and endogenous pacemakers. Maybe just tied up a little bit. A rhythm that lasts around 24 hours. We've got sleep-wake. We have any other ones than just sleep-wake? A testosterone cycle from L. Interesting. Okay, not menstrual yet. Core body temperature from Leah. Hormone, blood pressure. Good. Okay, I will also reveal the next section in a second. Please also tell me what an endogenous pacemaker is. Let's just do one at a time. What is an endogenous pacemaker? Oh, thanks for dropping in, Evening Rose. Yeah, we got an internal body clock that keeping our biological processes at time. We might have quite a few different ones, so we will pick up on one in a second. So what is an exogenous Zeitgeber then? If that was an endogenous pacemaker, what's an exogenous Zeitgeber? Internal factors that control biorhythms. Good. Is there a better word than control? Lower in there with entrain. Yeah, so we want to be fancy about it. We want to use a good term. We can use the word entrain our internal biological clock. So circadian rhythm lasts around 24 hours. Sleep-wake, release of hormones, body temperature, and blood pressure are all examples of circadian rhythms. Obviously, we're going to go into a lot more detail about the circadian rhythm. But the endogenous pacemaker, just for the term, we'll come up to it a few more times in this section. Any internal body clock that keep a biological process to time. An exogenous Zeitgeber, any external cue that sets or entrains an internal body clock, just shifts it around a little bit. Okay. Our next section is the sleep-wake cycle. So we do need to talk about this in detail. So just be aware of how this bullet point is written. Biological rhythms, circadian, infradian, ultradian, and the difference between these rhythms. So, it might be obvious what the difference is, especially the length of them. But we could be asked explicitly about the effect of endogenous pacemakers and exogenous Zeitgebers on the sleep-wake cycle itself. I will talk about them in passing in the others, but we do need to be particularly aware of their role in the sleep-wake cycle. Okay, no, we're not on the 5 stages of sleep just yet. SCN plus light. Okay, so people go into the evaluations really quickly. Not quite onto the evaluation yet. So what's the endogenous pacemaker for the sleep-wake cycle? Jay's put SCN, but I wonder if no one's typing that, is there? Okay, how does it work then? The SCN, what's it doing? Grace is saying melatonin. Does SCN release serotonin, or is it something else? So yeah, suprachiasmatic nucleus, pineal gland. So yeah, we've got a little bit of a loop there, haven't we? So we've got the sleep-wake cycle. The endogenous pacemaker is the suprachiasmatic nucleus. When it takes light, which the light in this case is the exogenous Zeitgeber, it's the external cue that's entraining our body clock, it sends a signal to the pineal gland, stopping the production of melatonin. But we also do have potential social cues as well when it comes to other EZs for the sleep-wake cycle, and that's maybe social cues. So we all tend to eat at certain points in time. Although interestingly, historically, that's not always been the same 3 meals a day cycle that we currently have now. And clocks as well. We're often looking at clocks. We're often aware of the time. If we're aware it's getting late, we might suddenly feel tired. If we feel like it's early, then we'll force ourselves to get up. So maybe we're altering our body clock with actual clocks and alarm clocks and stuff like that. Yeah, you can. I think you can probably get away with just SCN. Give it a go if you know how to spell it. Give it a go. If you spell it a little wrong, then it'll be the end of the world. Okay. So what is our piece of research that supports the idea that we have a sleep-wake cycle of 24 hours? Seafra. Yeah. His actual study is very complicated. If you ever read his original paper and the paper's about what happened, yeah, it's a bit weird. I don't think he necessarily finds what we say it finds, but that's all right. Just be simple. Oh, wow. Did I reveal all evaluations for this? Yeah, I guess so, actually. We've got Seafra. Let's see what you've picked up in there about Seafra. There we go. Guy lived in the cage for 6 months as details were given. Case study, not generalizable. Lights might have acted as an exogenous light giver, disrupting the endogenous pacemaker. Studies that controlled for artificial light found a 24-hour cycle. Thanks, Riley. That's really precise. And anything about unknown one? You've mentioned artificial lights. Anyone else got a study on artificial lights that we can use as a practical application? So going away from Seafra for a second. Okay, DXA's got Veta. Don't do diddly. Blue lights offer study. I think that's the better study. Okay, so let me run you through this. So our positive evaluation, we have Seafra. It's positive in the sense that it does seem this is a very extended period of time, 6 months, with no natural light. And also, obviously told the people who were looking after him not to tell him the time of day. So it was just seeing if he would keep to a cycle. It does seem that his internal body clock did maintain a cycle. And it was around 25 hours, which that's kind of significantly longer than 24 hours. So it almost seems like we do need natural light to entrain us. We don't have a perfect body clock. But could have been, maybe it was shifted by his use of artificial lights. Maybe that disrupted the endogenous pacemaker. And there are some studies out there, which I don't think they lasted as long as 6 months, though, did they? Found that if you control for that artificial light, we have a 24-hour cycle. Yeah, you did get lost about what day it was, didn't it? I guess if you're doing 24 hours days, it's shift like every 24 days. You would shift a day. So yeah, real world application. We know to avoid blue light in order to have a good night's sleep. I think that's a good application. So Veta found workers exposed to blue light shifted the timing of their circadian rhythm. This could help with practical applications. So if we know that blue light affects our circadian rhythm, maybe we could help us with jet lag. Maybe we can, I think you're on the plane. They turn the lights off a particular amount of time, and also shift work as well. You can get those light bulbs that go off in the morning that have a bright light maybe before dawn. Okay, I think we're good. On this. Let's move on, everyone, to the next rhythm. What is the rhythm, everyone, that lasts longer than 24 hours? The rhythm that lasts longer than 24 hours. Yeah. Okay. Oh, 1 or 2 mistakes, but more, maybe 3 mistakes. So longer than the rhythm itself takes longer than 24 hours to complete a cycle, and it is infradian. What is the term then? What is the infradian rhythm? 6 life goes in with menstrual cycle. Grace coming in with a seasonal depressive disorder, which is completely true. Okay. What, guys, is the endogenous pacemaker in the menstrual cycle? What is keeping people to this cycle. So Leah in there, yeah, with both of them. Estrogen and progesterone. Kira, you came with pheromones. That's not what I was after for the endogenous pacemaker. But then let's say, what is the exogenous IKBA and what's going on? Well, it's the argument anyway, because I don't think it's very well supported at all. Okay, so Ellis in there with pheromones. Not light levels. Not light levels for this. So Lola, pheromones of other women sync cycles. That's a great little sentence there, which means I can reveal what I've put. Infradian rhythms last longer than 24 hours. It's like seasonal depressive disorder, but what we talked about is the menstrual cycle. So in the about 28-day menstrual cycle, the level of hormones, estrogen and progesterone, act as endogenous pacemakers. So they are keeping the cycle to time. But it's argued that the pheromones of other women can act as endogenous IKBAs. Synchronization, so some will, if two people releasing the pheromones are near each other, one will lengthen their cycle, one will shorten their cycle, arguably to match. Okay. I think we're good. Which rhythm is this? This is infradian. All right, then give me the research support for this then, please. Let's have a look what we got. Stone McKintock, odor donor, but not supported by Trefan. This is what I'm going to go for. Something about lesbians not matching. That's the Trefan study. We've got the name. And we got Riley given a decent write-up there. Stone McKintock, 20 women, white pants from the armpits of other women on the top lip daily. Yeah, a little gross. Okay. And then we've got lesbians. The counter study was that lesbians who you would think, because there's just, there's two people living together that are sharing pheromones, you would expect them to be very much in sync, and they're not. So whatever was going on with Stone McKintock's study, maybe it was a kind of one-off. It is pretty gross. I wouldn't have volunteered for that study. Okay, so here's our evaluations. The criticism, potentially, that I haven't gone through is cycles can vary naturally. Some are slightly longer, some are slightly shorter. So you could have two people who seem to be separate in their cycles appearing to synchronize just because of natural variation. But I think the Trefan study is pretty good at kind of, if anyone you'd think would actually synchronize, you'd think it would be lesbians that live together. I don't think there's anything ethically wrong for this. People will have signed up to it. I don't know if you could really do demand characteristics, because you wouldn't know if someone's cycle was ahead or behind you, so you wouldn't know how to lie. I imagine they were told what they were wiping under their lip. Yeah, it's just gross. But things people do for science, eh? We are our final part of our final sections, everyone. What is the last rhythm? Ultradian. Excellent. Okay, so it's very easy to overdo this bit. I think historically, it's there's a lot of detail potentially we could use. But if we just wanted to get enough in there for an essay, last less than 24 hours is what we're going to go for. Stages of sleep, Leo, you are right. How long do these stages last, and how many times do we have them during the night, do you reckon? So we've got multiple times from Lisa Mellon, but you need to get a little bit more. On average, about 90 minutes. Alfie's got 4 or 5 times. Yeah. Okay, so let's just try and explain the stages in as few stages as possible. What is the first stage of the cycle? N1. I don't think we need to know too much about the waves, to be honest with you. So we've got N1, light sleep. Got alpha wave. Easy to wake up. Sense of falling asleep. Arena there of hypnic jerks. Sensation of falling. I would say just for each section, kind of have a little thing to say about each of the stages. Okay, tell me about the next one then. So we've got the lighter sleep of N1. What do we then go to, and what's happening with what's happening? So yeah, Zoe in there with N2. So it's a deeper sleep, harder to wake up. I wouldn't say N2 is light sleep, but it's not the deepest. Yeah, okay, complexes and spindles. Don't worry too much of all the brainwaves is a little bit much, but unknown one tells me my mic's bugging. I don't know if anyone else thinks it is, but let me know if it is. Anything else that's going on? What's happening with the body? Harder to wake, but what's going on with the rest of the body? Okay, it's fine for you. Okay, good. Yeah, it must be your computer. Decreased body temperature. Yep. Muscle relaxing, breathing slows. Yeah, good elements there. Okay, then what's the next stage? Stage 3. Okay, you're off there, dark humor. Good to have you on it. Stage 3, deep sleep with featuring delta waves. Slow wave sleep. But that's where I'm after. There is... Well, not quite a rapid eye movement, actually. Yeah, there is not 5 stages. Here's a funny story. So AQA themselves put up a glossary of key terms about lots of terminology, and they did put one out. There was one there about the stages of sleep, in which they said there was 5 stages and listed them. And then I got onto them being like, this has not been the definition for 20 years. Some of the textbooks have it, but like, here's all the research that's changed all of their perspectives about how we see sleep patterns. So we need to be up to date. And then they changed the definition on their glossary of terms to match what is actually true now, which is there's N1, N2, and N3. So if you've got 5 stages and you put it down, because in textbooks, I don't think you're going to get like any pushback there, but to actually be correct, this is how it is now. So yeah, you'll be fine. So let's go next stage, please. Next stage. Charlie, we could maybe put some of that in the evaluations, but I don't think we necessarily need to. You put down whichever you want. Okay, next one. Now, I'm not going to go stage 4. I'm going to say stage N1, N2, and N3, and then we get into REM sleep. Yeah. So REM sleep, we're just going to go say this, as you already were, dreams, but also we've got the paralysis element as well. Because, if someone's having like active dreams, they're running through like a forest or something like that, you don't actually want their muscles to actually engage and then start to do sleepwalking. But yeah, here we go. That's a little write-up of what we go. I've not gone deep into the waves on this because necessary, but you can add that if you want. Let's go then with what's the original like research then on the ultradian rhythms and sleep. That real world application is pretty good, actually. So we avoid waking up in deep sleep. I guess the reason those lights are pretty good to wake up to, because you can have those lights which slowly light up, and I think that allows you to kind of wake up when you're in light sleep rather than an alarm clock that just goes off like that. Kiteman, yeah, Dermot and Kiteman. What can you tell me about that? Okay, some good details there, but let me just run it through. Actually, this is my last slide. No, not quite. So Dylan Kaufman, EEG of 33 participants while sleeping. And basically what we're just saying is the brain waves followed a cyclic pattern, body relaxed and slow, I'll change it to slow, and high activation in REM. It just supports the stages. And we have already had the criticism, or one of the criticisms, but I'll probably just give you the next one for the sake of being able to finish the stream at a reasonable time, and that is there are significant individual differences. So yeah, someone in the chat mentioned newborns spend 80% of their sleep in REM, and obviously as you get older as well, it is variable. But there is an argument, because one of the I mentioned at the start of this section that the specification talks about the differences between the different cycles. We could come up with an argument to say that these things are potentially connected, because the longest part of the REM sleep often matches the lowest part of the circadian body temperature cycle. So maybe these cycles are in some way strongly connected. There might be different cycles, and they do seem to potentially use the same endogenous pacemaker, which is the suprachiasmatic nucleus. Okay. That, everybody, is the poster completed. So I hope that revision video was really useful for you. Just a reminder, I'm going to be doing some more of these over the course of the year, so do subscribe if you haven't already. And of course, you can get this digital version over on my website. I'll see you in the next one.